What's up, dude? You need five.
Whoa, ain't nobody got time for those dirty
But I
hands.
just cleaned them.
With what?
With my new hanky dandy Germex.
How do we know that cleans your hands?
They said it kills 99% of all dirt.
You can't believe everything in here.
But what if I just use soap?
What kind of soap?
Antibacterial soap.
How do we know which one's gonna clean your hands better?
Looks like we still got a new myth. Up top!
No.
Oh.
How does soap work? Well, soap has a hydrophobic water-repelling tail and a hydrophilic water-loving head. When you use soap, the hydrophobic tail is attracted to the dirt and oils on your skin, and the hydrophilic head attracts water.
This forms a soap micelle. Rinsing your hands will carry this soap micelle and any bacteria it's picked up off your hands.
The only difference between soap and antibacterial soap is an ingredient called triglycerin. While it's a perfectly effective bactericidal agent, it takes about 2 minutes left on the surface to do its stuff. And let's be honest here, who washes their hands for that long?
Hand sanitizer's active ingredient, alcohol, works by dissolving the cell membranes of bacteria. The only problem with this is that alcohol evaporates so quickly that there likely isn't time for hand sanitizer to be more effective. So what's our guess?
Plain old soap should be the best choice.
In order to test this myth, we need an appropriate environment for growing our bacteria. Since these bacteria will be from the surface of the skin, they won't quite be kept at body temperature, but we still need some way to keep the temperature constant and relatively warm. So we're going to build an incubator.
We will need this cooler, Christmas lights and tape, and a thermometer.
We started by taping 6 Christmas lights inside the cooler and monitoring the temperature. If it was too hot or cold after the temperature stabilized, we made adjustments.
Eventually, we had to cut it down to 1 light bulb. Any more and the temperature rose too high, potentially causing an explosion of bacterial growth.
Better set. We'll cultivate a test run or bacteria to make sure everything's just right.
First off, we prepared the agar. Bacteria are living things and they need nutrients to survive. The agar will provide this food. It's heated, poured into petri dishes, and left in the fridge to set.
To get our sample of bacteria, we're swabbing an unwashed hand, then it's into the incubator.
Three days later, there's no sign of bacterial growth. We're going to try to use a pre-poured agar plate instead and place it on a windowsill. Just to make sure we're getting a sample of a sufficiently germy surface, we're swabbing the.
That attempt failed, so we're making one last attempt, this time using a toothpick on the inside of a group member's mouth and placing the bacteria in a dish of pre-poured agar. This will go in the incubator.
Well, it's been another 3 days and there's still no sign of growth. We're going to have to call this one off and just focus on what went wrong.
Do you think it was sabotage?
Not exactly. With so many factors in the survival of our bacteria, it's not a huge surprise something went wrong.
Well, it seems like we did everything right. Food, warmth, and time. What else could have went wrong?
Well, the thing is, we didn't have all those things right. We couldn't get a hold of the agar that's specifically meant for bacterial growth, and the human stuff we had may not have been strong enough. The incubator was between 30 and 32 degrees Celsius the whole time, and that may have been too cold.
We got the pre-poured dish of agar. There's no way there's a problem with the nutrients there.
It's been a little cold out lately, and it's been pretty rainy, and the bacterial growth may have been too slow.
Alright then, I guess it's fair to say the myth is inconclusive for now.
Agreed.
Can I get the high five now?
Not a chance.